the topic of synthetic cannabinoids. It's a topic that evokes a lot of diverse opinions and ideas. In the cannabis space, many of those opinions carry with them negative connotations. I think many people in the cannabis industry, when they think about synthetic cannabinoids, they think of Marinol and its less than robust success as a synthetic substitute for whole plant medicines. The general public might think about synthetic marijuana. Stories in the press that we've seen over the past couple of years about these illicit street drugs turning people into zombies. The story of synthetic cannabinoids is very complex and it's very nuanced. It has ties to chemical warfare. It has ties to the discovery of a major physiologic system in the human body. The story includes a prominent organic chemist who's a descendant of John Quincy Adams and whose contributions to cannabis research have been largely forgotten. The story includes advances in chemical synthesis and biosynthesis, as well as illicit market substances that are named after, among other things, popular Japanese and Korean girl bands. But before we get to any of that, we have to cover some basic science. So to fully understand the story of synthetic cannabinoids, you have to understand a little bit about drug synthesis. And to understand drug synthesis, including the synthesis of cannabinoids, you have to have some understanding of chemistry. And obviously that's mostly outside of the scope of this workshop today. This is not a chemistry class. And I know that we're all bringing very diverse backgrounds and experiences. So for those without a science background, stick with me for about 10 or 15 minutes. We're just going to cover some basics. So typically there are three methods that you can use to produce cannabinoid extracts and isolates. You can do it through plant-based extraction. This is the process where phytochemicals are extracted directly from harvested plant material. And of course, this is the most commonly used method of cannabinoid production. You can use chemical synthesis and you can use biosynthesis. Now, chemical synthesis and biosynthesis can be discussed collectively under an umbrella term, which is synthetic production. And given that we're discussing synthetic cannabinoids today, we're not really going to talk much about plant-based extraction processes, except to provide some points of comparison. It's a fascinating area of cannabis science and production and product development, but that's another talk, maybe for another day. Today, we're going to really focus on synthetic production of cannabinoids. So chemical compounds are, of course, comprised of atoms, and they're connected by chemical bonds. Typically, the synthetic production of a specific compound will require that chemists break apart some of these existing bonds and form new bonds. And so when you have the synthesis of a complex molecule, something like a cannabinoid, it includes a considerable and varied number of required reactions. And often each step of the process focuses on the reaction of a single chemical bond. So chemists can use different methods to cause a reaction. So for example, some bonds might be influenced simply by applying heat. Some bonds might respond to the exposure of ultraviolet light. Others might react to things like electric currents. Essentially, what chemists are trying to do is they're trying to mimic the processes that occur in nature to construct chemical compounds through controlled reactions. So both chemical synthesis and biosynthesis are processes where controlled chemical reactions using two or more agents are combined to produce a desired compound. No plant material is required here. Essentially, these are cannabinoids that are created in a laboratory. Now, chemical synthesis involves two or more non-biological or non-living compounds. And these are combined in a controlled chemical reaction. And of course, the intent is to create a desired output. So chemists can start with two or more materials. Usually they combine that material with a catalyst to accelerate the process, and they create a more complex output as a result of that reaction. And they can perfect this process to create desired end products in large quantities, in short periods of time, and with identical chemical structures to their naturally occurring analogs. Now, broadly, biosynthesis refers to reactions that occur in a living organism. And typically these are reactions that convert simple structures into more complex structures. Biological systems have mechanisms to catalyze reactions, right? We have enzymes that can initiate and facilitate chemical reactions. And then we have, energy sources that are required to drive these reactions to completion. Biosynthesis in the context of synthetic cannabinoid production refers to the use of a biological or living organism, and they use those as the agents in the controlled chemical reaction, again, to yield the desired output, or in our case, to yield the desired cannabinoids. So biosynthesis differs from chemical synthesis in that one of the inputs used in the chemical reaction is a living biological organism. And usually that input is a very simple organism. It's something like a bacteria or a fungus, like a yeast or an algae. So these living organisms that are used in biosynthesis are kind of like the engine for the chemical reaction, because they produce their own enzymes to catalyze the reaction. So a simple example of biosynthesis is the process for alcohol production. So you've got sugar that you get from either grains or fruits. You combine that with water and yeast. The yeast metabolizes the sugar. It processes the sugar for energy. And that's the fermentation process that's happening. And then the byproduct of that fermentation process or the metabolism is carbon dioxide and alcohol. And you can see that here on the slide. This is a vat of wine must, and you can see the CO2 being released through the fermentation process. Those are the bubbles that are on top of the must. This is a really simple process with inputs that are naturally available, but you cannot produce cannabinoids. You can't produce compounds as complex as cannabinoids using only naturally produced products. So to produce cannabinoids using biosynthetic processes, you have to start with a biological organism that's been genetically engineered. So changes are made to the organism so that its cells produce the enzymes required to create the desired byproduct. This genetic modification kind of hijacks the organism's natural chemical processes to create the desired output. And DNA modification is difficult, it's expensive, it requires years of research and development to perfect, but it's happening right now. In this field of research, the engineering and modification of biological organisms and systems and processes is called synthetic biology, and this field is changing the manner in which commercial compounds are produced, not only cannabinoids, but also pharmaceuticals and other consumables and industrial items, and also wellness products. And currently, we see chemical synthesis and biosynthesis being commonly used in industries like the pharmaceutical industry for the production of drugs. So for example, yeast fermentation is used to biosynthesize insulin. Although I think that Genentech, when they initially created this biosynthesis for insulin, they used E. coli. Prior to Genentech coming up with this idea of biosynthesizing E. coli, insulin was harvested from livestock pancreases, mostly pigs and cows. We have another example like aspirin, which is acetyl salicylic acid. That's now produced entirely synthetically, whereas initially it was derived from chemicals found in the bark of willow trees. So just to put a kind of a final point on the chemistry of synthetic production of cannabinoids, I think it's helpful to consider a sort of very basic description of how cannabis plants produce cannabinoids naturally. So cannabis plants extract CO2 out of the atmosphere, and they use that CO2 to form sugar. And then the plant uses those sugars, and through a number of different enzymatic conversions, they eventually create CGBA or CGBV. These are the biological precursors to most cannabinoids. So you can see in the slide here, you can see that these enzymatic processes that convert glucose into CGBA. You've got on the left side, you've got glucose at the top that's converted into glyceraldehyde 3-phosphate, that's converted into pyruvate, which is converted into acetyl coenzyme A, and then the Krebs cycle gets involved in there. And frankly, unless you're a biochemist or a molecular biologist, you might not really understand what's happening on the left side of this slide. And that's kind of the point. the process involved in reactions to convert glucose to CGBA are complex. So when determining how to synthesize cannabinoids, chemists have to determine how to replicate these steps, starting with either non-organic or living molecules, or they have to determine where in the series of steps they can insert some genetic material to subsequently produce the same process to create the biological precursor to cannabinoids. So it's complex, but it's currently being done. We'll return to synthetic production of cannabinoids later in the discussion, but I just wanted to give everyone kind of some groundwork, some fundamental information about how drug synthesis works. I know that's kind of heavy on the chemistry. So I'm going to pause here just in case we have any questions before I move on. All right. So Western chemists they began exploring the chemistry of cannabis in the late 1800s, and some of the first chemists to do this were Thomas Wood and his colleagues, W.T. Spivey and Thomas Easterfield, and they began the work of identifying cannabinol (CBN) from an alcohol extraction of cannabis, and their research was later confirmed by a British chemist whose name was Robert Kahn, and he confirmed and published the structure of CBN in 1940. The American chemist Roger Adams was actually the first to isolate and synthesize a cannabinoid. Adams is a well-known and respected chemist. He was the department head of chemistry at the University of Illinois between 1926 and 1954, and he greatly influenced graduate education in America. He taught over 250 PhD and postgraduate students, and he also served as a scientist at the highest levels of government during World War I and World War II. And in 1939, just 2 years after Congress passed the Marijuana Tax Act in 1937, Adams received a Treasury Department license to work with cannabis oil in his lab. And shortly thereafter, his team first identified CBD in 1940, and he secured a patent for isolating CBD in 1942. So Adams was also the first to identify and synthesize THC, but he wasn't able to isolate THC directly from the plant. So he knew, obviously he knew there was a psychoactive component to cannabis, and he assumed quite accurately, it turns out, the molecular makeup of the cannabinoid, but the technology required to isolate THC from the plant, this was later used by Raphael Mechoulam and his team in the 60s. It just wasn't available to Rogers when he first started doing his work earlier in the century. So instead, he synthesized THC by converting the molecular structure of other cannabinoids, and principally CBD. So in fact, in 1940, Adams published a method for converting CBD to THC, and by 1949, he had synthesized an entire catalog of THC analogs, and these were the first artificial cannabinoids. And Adams would eventually publish about 27 studies on cannabis. And they were all published in the American Journal of Chemistry. And to this day, chemists still use his system to measure potency in cannabis, and it's called the Adams scale. I just want to take a moment to talk about isolation versus extraction. We're going to be mentioning some of these terms occasionally. So isolation is a separation technique. It's where we can obtain a purified compound. It's the key difference between extraction and isolation is that extraction is a technique in which we can separate a compound or compounds from a mixture, and isolation is a technique where we can use that's what we use to purify the extracted compound. Roger Adams is also connected to the American intelligence community's research on the control of human behavior. This began in approximately 1943, and it was specifically on methods to compel subjects to speak truthfully, even against their own will. So there were some declassified documents that were revealed at the Office of Strategic Services. This was the predecessor of the Central Intelligence Agency. They began highly classified experiments during the Second World War, and with some of these experiments conducted on unwitting subjects that measured the potential of a cannabis extract as a truth serum. And these documents appeared because somebody had them in a desk drawer, and the gentleman died, and his wife found them, and they were published. So they became declassified. Adams might have provided the THC-rich extract that was used to perform some of these experiments. And certainly the OSS relied on his work on cannabis chemistry to conduct these experiments. Now, and this is a copy of one of the documents. Lest I leave you with a negative opinion of Roger Adams, because that's not what I want to do. I want to relay a couple of anecdotes about him. First, he was no friend of Harry J. Anslinger. So, of course, those of us in the cannabis industry, we know Harry J. Anslinger as the father of the war on cannabis. Anslinger was the first commissioner of the Federal Bureau of Narcotics. This was the predecessor to the DEA. Anslinger fully embraced racism as a tool to demonize cannabis, and he used newspapers to distribute, in many cases, his own authored misinformation. And many of those articles included racial themes in which he would denigrate immigrants and communities of color. Roger Adams was publicly criticized by Anslinger. Anslinger apparently perceived Adams as having a bit too much enthusiasm for his work after Adams had reportedly commented in mixed company about how about the pleasant effects of cannabis. And Anslinger would publicly scold him, and he said, quote, "In my opinion, this drug is bad for human consumption and should be painted so." An enemy of Anslinger is a friend of mine. He also found himself in the crosshairs of FBI Director J. Edgar Hoover, who suspected Adams of being a communist sympathizer because Adams belonged to a group called the Lincoln's Birthday Committee for Democracy and Intellectual Freedom. This was a body of academics. They were opposed to Nazi pseudoscience and dominant race theories. In fact, historians would eventually state that Adams was an early anti-fascist. So I think it's time that the cannabis industry pay the proper respect and attention to Roger Adams that is commensurate with his contributions. So let me just pause here and see if we have any other questions. All right. So typically, cannabinoids that were initially synthesized for research and for use in research, they're usually named after the person who first synthesized them, or after the institution or company where they were originated. So, for example, you might see things like JWH synthetic cannabinoids. These are named after John W. Huffman. That's Professor Huffman pictured here. AM cannabinoids are named after Alexandros Makrianis. HU cannabinoids are named after Hebrew University. These are cannabinoids that were created by Raphael Mechoulam and his team at Hebrew University. CP compounds, those were synthetic cannabinoids that were made at Pfizer. It stands for Carl Pfizer. The structures and stereochemistry of CBD and delta-9 THC were further elucidated in Mechoulam's laboratory, CBD in 63, THC and delta-9 THC in 64. And so the research into the therapeutic potential of individual cannabinoids began shortly thereafter in the early 70s. A number of studies came out, anecdotal reports were starting to leak out about how delta-9 THC might actually be useful for suppressing pain. In 1979, a team working at the pharmaceutical company Pfizer developed a synthetic THC analog called CP47497. And this was very structurally similar to THC, and it had similar effects in animal models. So it was an analgesic, it was a motor depressant, an anticonvulsant, it had hypothermic effects. And so this group eventually would publish their research in 1982. Now, the Pfizer program was eventually terminated, but before they terminated, they created a whole bunch of compounds. And one of them was called CP55940. This was a really important synthetic cannabinoid because it would eventually help researchers discover the CB1 receptor. So Alan Howlett and her colleague, William Devane, I think in 1988, discovered the CB1 receptor by making CP55940 radioactive. And then they sort of they traced it in the body, and they used it to detect binding sites, and they discovered the CB1 receptor, which was instrumental in the discovery of the endocannabinoid system. I mentioned the word elucidation. So elucidation is the process of determining the chemical structure of a compound, identifying how a molecule's atoms are arranged in space, in three-dimensional space, can enable chemists to predict pharmacodynamic effects, how the molecule might react with other molecules, the boiling point of the molecule, which substances might metabolize the molecule. Now, today, the process is facilitated by nuclear magnetic resonance spectroscopy. But this is a technology that was discovered, even though it was discovered in the late 1930s, it wasn't really available to chemists until much later. So it wasn't available to Roger Adams, but was available to Mechoulam. And so they were some of the first chemists to use that technology to fully elucidate CBD and THC. A team at Eli Lilly and Company developed another synthetic cannabinoid. This one was Nabalone. And they began publishing results in the mid-1970s. So in 1985, the FDA approved Nabalone. It's a synthetic THC. And they approved it to mitigate chemotherapy-induced nausea and vomiting. Curiously, Eli Lilly actually withdrew Nabalone for approval. They cited commercial reasons. Maybe they thought that it wasn't profitable. Maybe they weren't comfortable with having a synthetic cannabinoid drug. But Valiant Pharmaceuticals would eventually acquire the rights from Lilly in 2004. And they sell the product now under the brand Sesamet, and it's a capsule that's administered orally. In other countries, Nabalone is also approved it's approved as a use for an antiemetic. It's approved for glaucoma, spasticity in MS, cachexia, and also as an adjunct analgesic for neuropathic pain. Starting in 1984, John Huffman and his team of researchers at Clemson University, they began synthesizing cannabinoids, and they were specifically looking for medicinal properties that were similar to THC. So they did their research over the course of 20 years, and Huffman's team developed over 400 synthetic cannabinoids that would be really useful in the study of cannabis pharmacology and providing researchers with a better understanding of endocannabinoids and also cannabinoid receptors. 1985, the FDA approved dronabinol. So many of you know this under the brand name Marinol. They approved it for the treatment of nausea and vomiting associated with cancer chemotherapy. Dronabinol is a capsule with synthetic THC and sesame oil. It's of course administered orally. In 1992, the FDA also approved dronabinol for the treatment of anorexia associated with weight loss in patients with HIV/AIDS. 1988, Raphael Mechoulam and his team at Hebrew University in Jerusalem, they created HU210. So this is very similar to THC. HU210 is a potent analgesic. It's a potent anti-inflammatory. Also, Mechoulam's research suggested that HU210 could reduce the buildup of beta amyloid proteins in the brain. These are proteins. They're kind of like a plaque that build up and cause inflammation, and they've been associated with the onset of Alzheimer's disease. More recently, Mechoulam's team announced a synthetic cannabinoid acid molecule. This is HU580. This is a CBD methyl ester molecule. It suggests greater stability and higher potency compared to phytocannabinoids. So the acidic cannabinoids, THCA and CBDA, they're really difficult to work with, especially in a research environment because they degrade so easily. So this cannabinoid, HU580, it will enable researchers to further explore the novel therapeutic approaches of the acidic cannabinoid-like compounds And HU580 has already demonstrated anxiolytic and antiemetic properties in animal models. And this is through activation of the 5-HT1A receptor, that's a serotonin receptor. So there are now hundreds and hundreds of synthetic cannabinoids. There's so many that they have like different categories depending on the structure. Some of them are THC analogs. Others don't look, the chemical structure don't look anything like THC at all. And more chemical analogs are created, and many of them are created to subvert the legal regulations that have been placed on earlier generations of synthetic cannabinoids. We'll talk a little bit more about that later. So we've talked about the chemistry of synthetic cannabinoids. We've talked a little bit about the history of synthetic cannabinoids, and we're about 30 minutes in, and we haven't yet addressed kind of a fundamental question, which is why? Cannabis plants are metabolic factories that produce scores of different molecular compounds, over 100 different cannabinoids alone. Cannabis plants can produce over 400 chemical compounds. So it begs the question, if cannabis plants are so prolific at naturally producing compounds, why do we need synthetic cannabinoids? Anyone want to take a guess? You have to unmute yourself if you want to take a guess. Why do you think initially we needed synthetic cannabinoids? Yeah, someone in the chat just said research. That's absolutely right. Initially synthetic cannabinoids were developed for research. Because there were legal restrictions, and it limited the availability of plant-derived cannabinoids, and it really forced researchers to develop similar compounds. More recently, however, businesses are starting to see profit-related reasons for developing synthetic cannabinoids. By the way, this is a microscopic picture of a trichome. So from a commercial perspective, the goal is to produce cannabinoids cheaply, efficiently, and reliably. And that can be really difficult to do if you're doing conventional plant cultivation. So first of all, cannabinoid production is not optimized during plant production. Cannabis plants produce cannabinoids only during a specific phase of growth. And those cannabinoids are concentrated only in the unfertilized female flower. And during most of the plant growth phase, the plant doesn't produce any cannabinoids at all. Furthermore, the cannabinoids are stored only in trichomes, and so these are fat soluble. The cannabinoids are fat soluble. They can't exist in other aqueous parts of the plant. In fact, in their natural state, cannabinoids are actually toxic to cannabis plants. And you can see on this picture, this is a picture of a bunch of trichomes on a cannabis plant, and they're mushroom-shaped sacs, and they have a stalk. And the stalk keeps the cannabinoids and other essential oils away from the plant material. When the plant matures, you've got an extraction process that's required to isolate the cannabinoids and remove all of the unwanted material, like chlorophylls and plant lipids and waxes. These are things that detract from the end product, detract from the user experience. And even though producers are getting really savvy about plant extraction, it still remains a pretty inefficient process. So while startup costs of biomanufacturing are quite high, in the near future, the cost of mass producing synthetic cannabinoids in something like a bioreactor, and when I say bioreactor, it might look like something like this, but it also might just look like a big tank or a big vessel. It'll likely be far cheaper than traditional cannabis plant farming. Cannabis cultivation, especially at commercial scale, is land and capital intensive. Greenhouses and indoor grow facilities at commercial scale can cost tens of millions of dollars. Biomanufacturing enables a consistency that's impossible to replicate in plants. Biomanufacturing is not influenced by weather or by pests. It requires less energy than indoor cannabis grown plants. And synthetic cannabinoids can be patented. And companies are really reticent to invest in the research and development of a therapeutic substance if there are no guarantees that their intellectual property won't be protected. So from a pharmacology perspective, synthetic cannabinoids offer researchers an opportunity to improve the potency, the affinity, and the efficacy of cannabinoids. So for example, many companies are looking to develop a cannabinoid that has very high affinity at CB2, but low affinity at CB1. And this is going to help consumers benefit from the therapeutic properties of CB2 activation. This would be like things like anti-inflammation and pain mitigation, but while avoiding the side effects of CB1 activation. And of course, when we talk about CB1 activation, usually we're talking about psychoactivity. From a medical perspective, synthetic approaches to cannabinoids can help facilitate the creation of products that include cannabinoids that rarely occur naturally or that are difficult to extract. And this offers new therapeutic possibilities. So in fact, there are some companies that are creating completely new cannabinoid-like molecules that are not even found in nature. So HU580 is a good example of that. Sorry, it's my cat complaining in the background. So remember that there are over 100 identified cannabinoids, and many have already demonstrated therapeutic possibilities, but many of the minor cannabinoids are understudied because of their rarity in natural plant production. So mass-produced synthetic versions of things like CBG, CBC, THCV, Delta-8, CBN, other cannabinoids, these are going to provide new opportunities for research, for product development, and novel approaches to treating disease. And we have companies now that are working in either the chemical synthesis or biosynthesis space. So a good example of a company operating in the chemical synthesis space is Naramco. They're a Delaware company. They're one of the largest manufacturers of controlled substances in the world. They supply major global pharmaceutical companies with compounds produced at their facilities. And their facilities are FDA approved and DEA approved and Health Canada approved. Basically they're a wholesale manufacturer and supplier of illegal drugs. Naramco currently makes pharmaceutical grade synthetically derived CBD that's 99.5% pure. And this typically meets the threshold for THC free products. And Naramco also has existing agreements to supply CBD to companies for the sale of pharmaceuticals in both Canada and Mexico. And they also produce synthetic Nabalone and synthetic dronabinol. In the biosynthesis space, there's also a number of companies who are attempting to produce cannabinoids from yeast and from bacteria and from algae. And all of these companies have they've developed a proprietary process, but they all the processes are all pretty similar. It all includes the introduction of genetic material to enable an organism, a simple organism, to produce the chemical reactions necessary for cannabinoid production. So for example, in 2018, the Cronus Group, this is a global cannabinoid research technology product development company. They announced a partnership with a smaller company called Ginkgo Bioworks in Boston, and they're going to develop cannabinoids from yeast fermentation. Another company, InMed Pharmaceuticals, they're located in Vancouver. They're focused on the development of cannabinoid-derived pharmaceuticals. They're developing a biosynthetic manufacturing process using fermentation with genetically modified E. coli bacteria, and they intend to use those cannabinoids for the production of their own drugs. We have companies now who are trying to genetically modify the cannabis plant itself to produce cannabinoids, for example, from shoot to tip. So for example, companies are looking at methods to produce cannabinoids outside of trichomes. This would substantially increase the cannabinoid yield. We have companies who are looking to try to add enzymes into cannabis plants to make cannabinoids more water soluble. This would make them less toxic to the plant, and water solubility also facilitates simple extraction processes. And it produces cannabinoids that might have broader uses in the market. And you think about, water solubility for, beverages and for food products. So in summary, the primary benefits of chemical and biosynthetic manufacturing are commercial scalability, precision and purity, even for rare cannabinoids, repeatability and consistency. And given the pace at which the industry is moving, and given the demand for products and the advancement in new product development, it's predictable that the industry is going to see a significant increase in the demand for these kinds of cannabinoid extracts and isolates. And in many sectors, the demand will not just be quantity. Some of that demand is going to require better standards of quality, production, specificity, especially when you consider derivative products, products in which cannabinoids are going to be used just as, one of the ingredients. You think about beverages and food and wellness products. So, consider, for example, companies like Coca-Cola or McDonald's. When they introduce cannabinoids into a product, and I guarantee you they will, they're not going to be sourcing their cannabinoids from a cannabis farmer in Humboldt County, right? They're going to require a massive amount of product. They're going to want the same product every single time with no variables, and they're going to insist on complete consistency and purity. Research in pharmaceutical products will require even higher standards of production and consistency. So this demand that's required by these markets will likely be met by synthetically produced cannabinoids. I'm going to stop here and take a drink of water and see if we have any questions. Don't forget to unmute yourself. It looks like we've got something in the chat here. What are the names of the companies you mentioned? I'll put a couple of articles in the chat at the end. I'll just paste them in, and they talk about a lot of the companies. Good question. See if there's anything else in here. Any other questions about why companies are interested in developing synthetic cannabinoids? I
have a question.
Go ahead.
Hi, my name is Felicia. What does purity mean in terms of synthetic cannabinoids?
Yeah, so when you look at a pure cannabinoid, like a pure CBD cannabinoid, an isolate, it just means that product has over 99%. It's all CBD, right? And usually the way that these things look is they're crystals or they're crystallized powders. So, when you get a cannabis product, let's say that you have a cannabis flower, you have maybe 20%, maybe 25% by dry weight of cannabinoids, of the trichomes that are still in the flowers. But then you have, 80% of that dry weight is other stuff. It's plant material. It's other, essential oils that are in the plant. When you have a cannabis edible, some percentage of that product will be cannabinoids, and then the rest of it will be, chocolate or bread or whatever the other ingredients are. So when we talk about purity, we're talking about a product or a substance that has a single ingredient.
Okay, thank you.
Any other questions? I certainly don't mean to suggest that I think that, or I think synthetic cannabinoids should replace plant-based extractions. I do think that there's going to be a significant place for synthetic cannabinoids in the future, and I think there's a significant place for them now. I don't think that they're going to replace plant extractions, but there definitely will be a place for it.
Timothy, I have a question.
Yes.
My name is Cindy, and it looks like that Coca-Cola product has already been developed, and when they're using a synthetic product, is it typically just like one isolate that they've been able to, synthesize, or is it, multiple cannabinoids in a synthetic product? Is it pretty much just one isolate at this point?
Yeah, usually synthetics are a single compound, and as to whether Coca-Cola has a cannabinoid product in the works, there's a lot of speculation about it. I wouldn't be surprised if we see federal cannabis reform in the next 12 to 18 months, and shortly thereafter, we see CBD products in something like Coca-Cola.
Yeah, I believe it.
Okay, any other questions before we move on? All right, so what does any of this have to do with synthetic marijuana? Remember that when we started out this talk, we had a slide that had zombies on it, and there have been a lot of articles in the press over the past couple of years, 10 years, about these synthetic drugs that are turning people into zombies. What's the relationship among synthetic cannabinoids and these illicit products that we hear about, like spice and K2? Well, recall Professor Huffman. He's a professor of chemistry at Clemson University in South Carolina. I just want to quickly review Huffman's work. He began researching synthetic cannabinoids in the mid-'80s, and again, they were focusing on synthesizing new compounds that would activate mostly the CB1 receptor. And he and his colleagues would eventually create more than 400 new synthetic cannabinoids. And naturally, they published their research, they published their formulas, they published their conclusions in peer-reviewed journals. In 2008, Huffman received a message from a German blogger who sent him an article from the German magazine Der Spiegel about a German man who was using a JWH compound, it was JWH18, as a recreational intoxicant. And Huffman would subsequently learn that multiple commercial products based on his compound started to appear in Europe in around 2006, after Huffman had published a paper describing how to make the compounds. In fact, he's somewhat notoriously quoted now as saying, JWH18 can be made by a halfway decent undergrad chemistry major in 3 steps using commercially available materials. So it was diverted into the illicit market. Now, typically, the packaging for these products, they look something like this. They usually list only natural herbs as ingredients. They often have, quote, not intended for human consumption, wink, wink. Of course, to clarify, they absolutely should not be consumed, but the language on the product was initially used to provide some layer of legal protection. Early products were marketed as incense, herbal blends, potpourri. They had names like Spice Silver and Spice Gold, Spice Diamond, Yucatan Fire and Smoke, 8KB48 and 2NE1. These were the names of popular Japanese and South Korean girl bands. These products were often mixes of herbs that included things like blue lotus and lion's ear, honeyweed, beach bean. And a lot of the products claim that it was this mixture of herbs that can produce these cannabinoid-like effects. But subsequent testing would reveal that these products contain synthetic cannabinoids, including multiple cannabinoids created by Huffman, compounds initially invented by Pfizer, compounds invented by Mechoulam and his team. And because many of these compounds were structurally distinct from THC, they didn't look like THC, but they still were binding with the CB1 receptor. Initially, they weren't really addressed by the Controlled Substances Act in the US. They weren't illegal initially. And they provided people with an alternative. It was an alternative for people who wanted to avoid the legal consequences of cannabis use, or an alternative for people who needed to pass a drug test. Now, the DEA has subsequently closed this loophole. They now state that any synthetically derived tetrahydrocannabinol remains in Schedule 1, and they specifically address synthetic marijuana. They describe it as a mixture of plant material with any synthetic psychoactive chemical sprayed onto it. There are now hundreds of these products available commercially. They're almost all through the illicit market. New and inventive methods for consuming these products continue to appear. There was one story about prisoners in Great Britain who were receiving pictures drawn by their children that had synthetic cannabinoid mixtures sprayed onto them. And so after they would get them into the jail cells, they would actually smoke the paper. But again, why are these illicit market products turning users into zombies, right? This is a real website about, these are sort of a public marketing campaign about the dangers of using these products. We have FDA-approved synthetic cannabinoid medications that are currently being prescribed, right, by doctors to patients. So what's different about these illicit market substances? The first thing to note is that the adverse effects of any synthetic cannabinoid can be much more severe than those associated with phytocannabinoids, cannabinoids from the plant. THC from the plant is a partial agonist at CB1 and CB2, so it only partially activates the receptor. It doesn't activate CB1 to the fullest extent. Most synthetic cannabinoids are full agonists with high affinity at CB1 and CB2, and full agonists can create higher risks of very severe adverse effects. So for example, HU210 is highly potent. It has a high affinity at the cannabinoid receptors when compared to THC. It's been studied in primates. Researchers determined that HU210 has a longer duration of action than THC because it has a very slow rate of dissociation from the cannabinoid receptors. In fact, the duration is so long that it's referred to as pseudo-irreversible. So HU210 gets in the system, it binds to CB1 receptors, and it just doesn't let go. When HU210 is administered intravenously, it has a duration of 1 to 2 days. And you compare that to THC, has a duration of maybe 5 to 7 hours when it's administered intravenously. And researchers have also performed intraventricular administration. This is direct administration into the brain of HU210, and this route also produces effects that last at least 24 hours. And based on the duration noted collectively for intravenous and intraventricular, they're speculating that the prolonged duration of HU210 is not a product of an active metabolite. It's just because it has such a great affinity for CB1. So when we eat THC, for example, it gets converted in our liver to 11-hydroxy, and it can last 6 to 8 hours. That's because of the metabolite that's created. HU210 is just because it holds onto that CB1 receptor and doesn't let go. All right, we're going to put our chemistry hats back on just for a couple of minutes here. There might also be a few other reasons, so a few different reasons why synthetic cannabinoids have higher binding affinity to CB1. So for example, there are several synthetic cannabinoids that have a 7-carbon side chain. Delta-9 THC is, you can see it on the slide here, is right in the middle. Delta-9 has a 5-carbon side chain. Can you guys, if I put my cursor on the screen, are you able to see it? Great. So a 5-carbon side chain, this is the side chain. And if you count the carbons, it's 1, 2, 3, 4, 5, right? But if you look at these synthetic cannabinoids like CP55940, you have 7, 1, 2, 3, 4, 5, 6, 7. Nabalone over here in the bottom right, 1, 2, 3, 4, 5, 6, 7 carbons. HU210, 7 carbons on that side chain. The length of the side chain is believed to influence a compound's pharmacodynamic effects, and synthetic cannabinoids like HU210, 55940, the ones that have the 7-carbon side chain, they demonstrate far higher affinity to CB1 receptors than Delta-9 THC, and they're far more potent than Delta-9 THC. And it's suggested that Nabalone, for example, has a binding affinity at CB1 that's 30 times stronger than THC. HU210 is somewhere between 100 and 800 times stronger at CB1 than THC. In fact, until recently, it was thought that all naturally forming cannabinoids were limited to at most a 5 carbon side chain. But in December of 2019, there was a team of Italian researchers that announced the discovery of 2 new cannabinoids, and we're still discovering new cannabinoids. But these were called THCP and CBDP, and both of them had a 7 carbon side chain. And just like the synthetic versions, THCP binds to CB1 in vitro, at least, in a manner that's similar to some of the other synthetic cannabinoids. It's a potent full agonist at CB1, and it's thought to be about 30 times more potent than THC. When we talk about affinity and potency, sometimes they're used interchangeably, but they're slightly different. Affinity for a receptor is the extent to which a drug binds to the receptor at any given drug concentration, and the firmness with which the drug binds to the receptor. When we talk about potency, we're really talking about a measure of the necessary amount of a drug to produce a specific effect. So, when we're talking about affinity at CB1, we know it's going to produce that pharmacodynamic effect of psychoactivity. So we can also kind of talk about the potency there as well. It's somewhat interchangeable at CB1. So there are that's not the only, the 7 carbon side chain is not the only possibility for, the reasons why this sort of high binding affinity exists. So for example, THC has a methyl group that's a carbon with 3 hydrogens at the carbon 11 position. And Nabalone, for example, has a double oxygen bond at the carbon 11 position. So synthetic THC analogs that have that double oxygen bond, or maybe a hydroxy group, that's an OH, attached at carbon 11 tend to exhibit enhanced activity and higher binding affinity at CB1. A good example of a compound that has a hydroxy group at carbon 11 is 11-hydroxy THC, and that that has a much higher affinity at CB1 than delta 9. If you're confused about what carbon 11 means, it refers to the manner in which carbon positions are numbered in a cannabinoid molecule. So when we reference cannabinoid molecules, the numbering system is called dibenzopyrin numbering. You don't need to remember that, but you can see an example of it here on the right-hand side. So you can see delta 9 THC. This is delta 9 THC, by the way. It's called delta 9 THC because there's a double bond at the carbon 9 position, and that's the position that's circled with the red circle. Delta 8, for example, it's the same molecule as delta 9, except it has a double bond at the carbon 8 position. So there you go. In older scientific literature, you might also see references to delta 1 THC. This can be confusing. Does anyone want to venture a guess at what delta 1 THC is? It's the same thing as delta 9. Delta 1 THC is it's simply a reference to an older numbering system, and that's the one that's shown on the left. It's called monoterpenoid numbering. But you can see these are both THC molecules on both sides, and the numbering is just slightly different. So if you're reading older scientific literature and you come across delta 1, it just means delta 9 THC. A couple of people have just joined. For those of you who joined at 5 o'clock, we actually started at 4 p.m. Pacific. I think there might have been some confusion because some of the notices were 4 p.m. Mountain Time. We are recording this, so you'll have an opportunity to watch it. We'll send an email out to everyone about how to see the recording. In 2013, the National Forensic Service in South Korea reported that 90% of all seized synthetic cannabinoids were fluorinated, and they had no instances of fluorinated synthetic cannabinoids reported in 2010. So this is another example of why some of these synthetic cannabinoids might be causing some more of these adverse effects. Fluorinated synthetic cannabinoids are like 2 to 5 times more potent at CB1 receptors than unfluorinated analogs. An example of a fluorinated synthetic cannabinoid is AB fubinaca. This was developed by Pfizer in 2009 as a potential pain medication. It was discovered in synthetic cannabinoid products in Japan in 2012. The cannabinoid is responsible for, AB fubinaca is responsible for 2 instances of mass overdoses involving hundreds of hospitalizations. There was an event in the United States in 2018 in Connecticut, and then there were 2 in New Zealand in 2018, one in Napier and one in Christchurch. So very potent effects and very potent adverse effects. And chemists are continuing to find methods for increasing the potency of these cannabinoids. And with higher binding affinity to CB1, of course, comes greater risks of adverse effects. So, these reported adverse effects include agitation and coma, toxic psychosis, cardiovascular problems, respiratory depression, acute kidney injury, addiction, and even death in some cases. And sometimes the death can be from hypothermia. Recall that CB1 modulates our body temperature. And so it can reduce our body temperature. And if you have a full agonist at high concentrations, it can actually cause hypothermia. Except for a very few number of synthetic cannabinoids that are approved by governing bodies for patient use, these compounds were never intended to be used outside of research settings. And these products, because they're mostly circulated through the illicit market, have very little regulatory oversight. They have, the products contain extremely high dosages, in these illicit products. The material that they're sprayed on, they're not safe for human consumption either. But as long as cannabis remains criminalized and as long as it remains a Schedule I drug, people are going to continue to seek alternatives, especially if they're attempting to treat conditions that impact quality of life. And they're going to continue to make compromises and poor choices. And the only path forward, I think, here is full decriminalization, full legalization, and at the federal level. We need federal cannabis reform in order to address the situation we have with synthetic cannabinoids in the illicit market. So what are synthetic cannabinoids? Are they prescription medications? Are they research tools? Are they chemical weapons? Are they compounds in genetically modified plants? Are they dangerous illicit market products? Are they the future of cannabis medicines? I think the industry has to accept the fact that synthetic cannabinoids are probably all of these things, and they will continue to influence cannabis research and product development and public opinion in the future. I just want to remind everyone that Radicle Health has an array of curriculum options at a range of price points for students and working professionals. So whether you're helping customers make good product choices or treating patients or advocating for social change, or maybe you're just using cannabis yourself, you need evidence-based, factual information about cannabis. Radicle Health curriculum's up to date, it's comprehensive, and it's informed by both clinical expertise and instructional design expertise. And before I open it up to further questions, I also want to acknowledge the programs that the Community College of Denver will be creating for their students. They've just been approved to move forward with the design of 2 undergraduate cannabis programs, a Bachelor of Applied Science in Cannabis Science and an Associate of Applied Science in Cannabis Business. I've seen some of the proposed curriculum for these degree programs. It's very exciting, it's very progressive, and I think it's going to provide students with some very marketable skills and knowledge for entry into what we all recognize as a very exciting and dynamic industry. Again, for those of you who joined at 5, my apologies. I think there was some mix up in the time. We started at 4 p.m. Pacific, 5 p.m. Mountain Time. We are recording this, so we will post the recording. I thank you for your time today and for indulging me this afternoon. I think we have some time for questions, and I'll turn it over back to Dr. Obidi or to Ruth Ann.
Thank you, Timothy, for sharing your expertise with us today. I personally enjoyed it so much because you were speaking my language, the chemistry language. So thank you. Timothy will be delivering a second workshop on cannabis and the cardiovascular disease. This is scheduled on March 24th at 5 p.m. Mountain Standard Time. And I'm putting the link if you are interested. You can use this link to register. In addition, and in collaboration with the subject matter experts from the cannabis field, we will be delivering 2 additional workshops. One of them is on March 11th, focusing on talent acquisition in the cannabis industry. And the second one is scheduled on April 7th, and we are hosting a workshop facilitated by the CEO and Executive Director of LEAF 411, and she will be talking about making data-informed decision around cannabis usage. As Timothy said, those workshops are in preparation for 2 new degrees that we will start offering in the near future, and there are 2 of them, an AA, an associate in cannabis business, and then a bachelor's of applied science degree in cannabis science and operations. I'm gonna put my email address. If you have any questions about any of those degrees, please feel free to email me. Thank you everyone. Any questions for Timothy?
Are you going to be putting the links in for your upcoming, the ones that you just mentioned that were coming up in March and April?
Yeah, so if you click on this link that shows the cannabis cardiovascular disease, this will take you to the website where you will see all of them. So you will be able to register with this link too.
Great, thank you.
Mm-hmm.
I had an opportunity to attend the previous workshop. It was excellent. It was about opportunities in the cannabis space. So these are well worth your time if you have an opportunity to attend. Any other questions before we sign off this evening? Don't be shy. Okay, well, again, apologies if there was some confusion about the starting time. We did start at 4 p.m. Pacific, 5 p.m. Mountain Time. We did record this. I'll be posting it and sending out an email to everyone who attended tonight, and we'll get you the link to the recording. Thanks again to Community College of Denver, and thanks for everyone for spending your time with us this evening. And maybe we'll look forward to seeing you at a future workshop.